already or are interested in the near future in generating RAD data? Yeah, so almost everybody, okay? Again, we generate a lot of RAD data, and we really love the protocols that are presented in this paper. So there's a lot of RAD protocols out there. They have pros and cons, but we are using these protocols. And so we'll talk about them a little bit. And this is a great opportunity for you to ask questions if you're interested in some of the details, molecular details, as well as whatever else. I don't want to spend a huge amount of time discussing the different protocols and which ones are the best and all this stuff, but I want to sort of talk in general about some things. Okay? So this paper was my student Omar led this paper, and it's a method basically called Rapture. But the title doesn't really do justice to this paper, because actually what's more useful about this paper is the new RAD protocol that we develop in it. So we develop a new RAD protocol in this paper, and we add onto that RAD protocol a step of applying capture to RAD libraries. But in reality, we use the RAD protocol alone without capture more often than we do Rapture or RAD capture. So in general, I want to talk about both of those things. If people have interest in some of the details of Rapture and applying capture methodology on top of RAD libraries, I'm happy to answer questions about that. But I want to start with this, a couple paragraphs in here. So basically in this paragraph here, we talk about how there's sort of this spectrum of data resolutions. You can collect a whole genome sequence data, so you can try to interrogate every position in the genome in every individual. That's sort of one extreme. And you can also interrogate a single site in an individual. And for different questions, you need different amounts of data. So if money didn't matter, there is no reason to not sequence the entire genome of every individual. Unfortunately, because even if you sequence the entire genome of every individual, you can just look at a small subset. You can look at one locus, one site from that data. However, the more sequence costs money, library preps cost money, and the more data you generate, the more expensive it is. And so depending on your budget, and depending on the kind of questions you ask, we apply a method somewhere along this spectrum from whole genome sequencing to a single locus, depending on what the questions we have and what our budget is and these kinds of things. Okay? So we're interested in having molecular methods that work across this whole spectrum. So the whole genome sequencing is one thing, and RAD gives you a relatively high number of loci across the genome. You can use different restriction sites, and you can get millions of SNPs or 100,000 SNPs or something. But we wanted a method that was more in the range of thousands of SNPs, but could be applied really cheaply. Like we were looking at, our goal was 1,000 SNPs for $1, sort of on that range. $1 per sample for hundreds to thousands of SNPs. And that's why we ended up developing this method, Rapture, or RAD capture. And the basic logic behind it is we use RAD libraries. We use restriction sites to isolate these RAD tags that flank restriction sites. And then we further subset the genome by having these capture probes that further isolate specific RAD tags. Okay, so it's like a two-step complexity reduction process that is both somewhat random, depending on where the restriction sites are, but the capture step allows you to be targeted and target specific sites with specific sequences. Okay? So here's a question for some people is, so if you're going to do capture, why not just create whole genome library preps for all your individuals and then capture off of your whole genome library preps? So why do RAD and then capture? Does anybody have any idea?
Is it easier to multiplex? To isolate the RAD loci?
Yeah, it turns out that one of the beauties of RAD, and one of the reasons we do it a lot, is because you can generate libraries incredibly easy. So what happens is you digest genomes. So you have your DNA, extracted DNA, you squirt some restriction enzyme in there, and it cuts up the genome. And then right after that, you ligate on these barcoded adapters. So very, very quickly in the protocol do you get each sample individually barcoded. And then immediately after you've ligated on those barcoded adapters, you pool all the samples. So we always make RAD libraries in 96-well plates. So every sample has an individual barcode on it. And immediately after we've ligated these barcodes, we collapse 96 wells into one tube. And the majority of the library prep is gone through in a single tube. So you very rapidly collapse, say, from 96 well plates, 10,000 samples, into just 96 individual tubes. So you could collapse 10,000 samples into one 96 well plate really rapidly. They're all individually barcoded. And so the library prep process for RAD is much faster and cheaper per sample to get samples individually barcoded than whole genome libraries. On the order of like 50 cents or something. Okay? So that's our main motivation was we wanted to couple RAD with Capture because RAD just gives you a really cheap and efficient way to prepare individuals to generate libraries, basically. But it didn't give us a small enough number of loci. Maybe you could use a combination of enzymes or something, but Capture allows you to target specific loci. So we can do rapid, make libraries from thousands of individuals quickly using RAD, okay? So that's sort of the motivation for this Rapture method. And that filled in this niche of, for us, let's say one SNP, whole genome sequencing. Sort of RAD was in here. Let's say RAD with the way we do it in 6-cutter. So, maybe one SNP, a couple thousand SNPs, 50,000 SNPs, 2 million SNPs, the whole genome. These numbers depend on genome size and this kind of thing. But using these different methodologies, we have a pretty nice continuous resolution of genetic information depending on, again, the question and the budget for samples. Okay? So we've been doing RAD for a while, and so we figured out, okay, we're interested in this Rapture method to sort of fill in this gap here again, but we tested it and we had this issue. We talk about this in this paragraph. So we say, our initial goal was to investigate the potential of Rapture as a flexible and efficient method for sequence-based genotyping. However, our initial Rapture results contain very high PCR duplicate rates. So that would mean fragments, we isolate fragments from an individual, they end up getting duplicated in a PCR, and we sequence these PCR duplicates as opposed to individual sampling events from the original genome. So why were we concerned about PCR duplicates? So why do we not want to sequence, or why do we not want to just sequence PCR duplicates, and how do we detect PCR duplicates in our RAD data? So there are two important questions. Yeah, so I'm asking too many questions to actually be able to answer them in any way that makes sense, sorry. But basically, it's this. This is a simple way to think about it. Let's say that I had 10 sequence reads that all came from the same allele. So I knew I had a CG polymorphism at a particular site. And my data was 10 reads that were all C. If I calculate the likelihoods in the way that we did here, I would say, well, and I convert it into posteriors, I'd say the probability of a homozygous C genotype is like 0.999 or something, okay? And the probability of a heterozygote is very, very low, and the probability of the other homozygous is 0. However, what if the situation was actually that I sampled one C allele, then duplicated a whole bunch of times and sequenced all those duplicates? If I was to recalculate the likelihoods under that scenario, the likelihoods would be, like 0.7 and 0.3 and 0, not 0.9, the likelihoods and then the posteriors would be completely different. Because actually I had a 50% chance of sampling that one C, and then that C got duplicated many times in sequence. So the genotypic information, when I try to quantify my belief in the genotype of that individual given the data, if there are PCR duplicates, I think I have a really confident homozygous genotype, but actually I don't at all. The individual could have easily been heterozygous. So and this relates to the assumption, so taking the joint probability across those reads, the AND rule, multiplying the probabilities of individual reads, we're assuming independence, and if that independence doesn't exist, there's a big issue in terms of if there's a lot of clones, we think we're highly confident in our genotype, but we're actually not confident at all. Do people generally understand what I'm talking about there? So how do we detect clones in our sequence data? This is a really important question. Yeah, so basically, when we do the data, we use paired-end sequencing when we generate this data. We have a cut site here. All of the first reads start right after the cut site. But each one of these fragments that we sequence, it's a fragment of DNA that maybe is around 500 bases. It has a second read. And the second read, although the first reads all start in the same spot that's determined by where the restriction site is, there's a random shearing step in the protocol, so the second read starts at a random location, right? So in this case, these second reads started, this is the second read that's paired with this first read, started here, this one started there, this one started there. But sometimes what we see is situations like this, where the second read starts in the exact same place in these two supposedly different sequence fragments. The probability of this happening randomly is very low, right? We're shearing across, say a few hundred bases, and we're only sequencing 10 times. So we use this to infer that these are PCR duplicates. So one original molecule got duplicated and sequenced multiple times. So when we make RAD libraries, we can identify the extent of clonality or PCR duplicates in the data by examining the position of the second read. And there's software that automatically does that for us, and we're going to use that software in just a few minutes. Okay?
But you have to do paired-end sequencing. You couldn't do it with single.
But yeah, now there's new methods. So this was one of the big advantages of doing RAD in the way that we do it. Was this is the case. There's different types of RAD. You can do this so-called double digest RAD, or you can shear. There's new methodology that incorporates, so the issue with double digest RAD is that the second read is determined by a restriction site too. So all of the second reads start at the exact same site. However, there's new methods, I'm not up on the details of them, but they basically incorporate a random oligo that has a random, like a fragment barcode that basically allows you to determine if reads are clonal origin using a double digest method, but it does that by incorporating this randomer into the sequence. And exactly the molecular details of that, I don't know. But there are ways now to identify and remove duplicates using a double digest RAD approach. Okay? And theoretically, you could use those same methods with a single. For us, we actually utilize this information too. We interrogate SNPs out here in addition to SNPs covered by the first read. And let me just say this. If you've generated double digest data, it's fine. It's not, and you can't remove clones. You can work under the assumption that there aren't clones there. And if the library was very high quality, the clone rate is probably relatively low, but it's unfortunate, you don't know, and so there are situations where you can think you're very confident in your genotypes, but you're not because you really just have a lot of clonal reads. So in my experience, depending on the quality of the DNA that you start with and the particular protocol, the quantity and the quality of the DNA that you start with and the particular protocol you use, that has a large influence on the rate of these PCR duplicates in your data. So sometimes PCR duplicates aren't there at all, and so it doesn't really matter if you remove them or not. But other times they can be pretty high. So just be careful. And there are, you actually expect, for example, to have an excess of homozygous calls, an excess of homozygous genotype calls, if you have highly clonal data. So there are ways to look at your data and figure out, if you can't remove clones because you use the double digest approach, there are sort of ways to get at the potential clonality in those data. Okay? So, does anybody have any questions in general about, since like pretty much everyone in here is interested in doing RAD or plans to do RAD, does anybody have any questions about this? Or just briefly different protocols, different approaches? Yeah?
This may be a side question, but you won't have to use parent reads to do RAD or Rapture. There's no way around that.
Yeah, to do best RAD, we casually call this protocol. There was this, never mind, I don't want to bash anyone else's RAD protocol. I'm about to, and I'm just not going to. But anyway, just as a joke, we started calling this protocol best RAD. The one of the details about this protocol is the cut site can be randomly on the first read or the second read. So using this methodology, the protocol presented in this paper, actually you do need to do paired end reads, or half of your reads will not, so there is that. That is, for us it doesn't matter because we use paired end sequencing all the time. I highly recommend paired end sequencing. Paired end sequencing is not much more expensive than single end. It's not like you pay double the price for paired end. I think you pay 25% more or something like that. So we do everything paired end all the time.
Can I just also comment from conversations I've had a lot about this, trying to be as cheap as possible and figuring out where to cut corners, is when we work with sequencing centers that work a lot with biomedical applications, they push single end a lot, not realizing when you don't have good reference genomes and things, that it's also really helpful to have paired ends and other data. So I'm assuming a lot of people in this room don't have perfectly annotated genomes.
Yeah, so there are a lot of advantages to paired end data. We've actually worked with some sequencing centers where they only do paired end sequencing. But yes, it probably depends on the place you're sequencing for the different rates between paired end and single end. And I'd say more people do, my feeling, at least in my world that I work in, more people do paired end. And so sometimes it's hard to get single end data because you have to wait for a single end lane, everything's in flux with the number of lanes and depends on the machine and these kinds of things. But in general, we do paired end 100 or paired end 150 for almost everything. Yes, Gordon.
Just wondering from you first, Mike, maybe Paul and others, what's known in published literature? What's been published? What PCR duplicate rates have been published? What's known? What's been reported? First question, and then what's the effect of that on any downstream summary statistics or estimations? And has that been any?
Yeah, so we published right here 90%. Well, we don't show the data. We've worked, it's highly variable. So most data that's out, most RAD data that's out there right now, you can't quantify the duplicate rate in it because it's single end, and so it's unknown. In my lab, it's been extremely variable. It just depends on the quality and the quantity of DNA. And oftentimes, we approach a new genome, especially plants. Plants have you can do a theoretical prediction of the number of sites. You can assume a genome is random, and all the bases are equally represented. And you can predict the number of restriction sites that are going to be present in the genome. And when we do that calculation on animals, it's I'm amazed every time. It's right on. What we see in the data is exactly what we predict. But when we work with plants, it's all over the place. And so you have this issue where you can have a huge plant genome and you can think you have many, many sites, and you can put in a particular amount of starting DNA, and it turns out there's very few fragments in there, so you don't isolate very much. And then you have highly clonal data because of the genome size or the relative number of cut sites in the genome and these kinds of things. So it's hard to explain too because, Gordon, the more you sequence, the more clones you see. So the more fragments you sample, the higher the probability that you're going to sample a fragment that you'd already sequenced too, and this kind of thing. So you can't just, it's very hard to quantify. It's hard to generalize that. Because quality, quantity, genome size, sequencing depth, all those things influence the rate. As far as summary statistics, the best way to think about it is just the math that we did. You can see how much the assumptions that went into those likelihoods we calculated, and if that assumption of independence is very strongly violated, then the likelihoods that you calculate under an assumption of independence are virtually meaningless. So in terms of calling genotypes, your called genotypes are going to be very off, but if you call genotypes across the individual and any given genotype is off, your overall allele frequency estimate in the population may be reasonable. So it depends on what summary statistics. So it's hard to generalize these kinds of things.
So you said very variable in your lab. What's the range of PCR duplicate rate, the average?
Well, like 90% to typical, about 20%, I'd say.
90% of your sequencing effort goes to PCR duplicates?
Yeah, it goes to PCR duplicates. And it could be a lot lower, but it's a trade-off because if we went lower, we'd have to have we'd input more DNA. We'd probably use a higher reaction volume, and so we'd lose money on the amount of enzyme that we put into the, so for example, we do rad preps in really small volumes, like 10 microliters. And if we do rad preps and everything in 10 microliters, we have a relatively small amount of enzyme and everything that goes in there. Now, the advantage of that is it's very cheap. We put like a 0.1 microliters of a restriction enzyme into a digest, and hardly any ligase. And so the per sample cost in a library prep is nothing. It's virtually nothing. However, we're going to have higher clones than if we would have put in 5 times as much DNA, and we would have done 5 times larger volume, and more of the enzymes, and all these things. We would have had less duplicates. So it's a trade-off. For us, especially for doing something like Rapture, the sequencing is the cheap part, the library prep is the expensive part. But if we're doing something like PST1 rad with a 6-cutter, the sequencing is the expensive part, and we would prepare libraries with more DNA and be more concerned about trying to remove clonality because we'd be dedicating a lot more money to the sequencing and we wouldn't want to lose a lot of sequence. So even answering that question, it's better to, rather than to just make general rules that are kind of meaningless, it's better to understand the process and be able to think through it yourself in your given situation, I would argue. Yeah?
Weigh in on this. One thing that wasn't mentioned was talking about assembling anything de novo. So the most powerful analysis requires some sort of reference genome or something like that. It doesn't have to be a well-established reference, but it needs to be something. In that circumstance, and I don't think I touched on this, certain assemblers have certain expectations about the depth at a given site, and that depth can either be PCR duplicates, so hopefully you're removing that or accommodating it some way. But if your coverage is much greater than what the algorithm expects, you would start fragmenting your genome. You might imagine certain scenarios where, depending on what type of analysis you want to do, breaking things up in smaller and smaller pieces, not being able to place those reads in an appropriate genomic context can actually result in biased estimates of one way or another. So one thing that I like to use as an example for this, if you're doing whole genome sequencing data and you sequence something that's say 30x coverage, so your whole genome average of 30x coverage of each position, that might assemble pretty well, depending on which algorithm you use it. If you want to reconstruct a mitochondrial genome, because the mitochondria is in such high duplication rate in cells anyway, coverage might be 1000x. So now what that means is that even after you dedupe it, and you say you bring it down to an average of maybe 150x across all possible sites of the mitochondria, anything that's maybe even 80x, 90x, the assembler is going to think that something weird is going on there with that dramatic shift in coverage, and it's going to start fragmenting. So it's worth either using algorithms or explicitly incorporating that information that accommodates the sorts of situations. But it's really, really important to keep duplication rates and even average sequence coverage into account when you're trying to do anything.
Yeah, so it's definitely, I've run into that issue with certain assemblers. Some assemblers deal with coverage, variation in coverage much better than other assemblers. It totally depends on what you're trying to do, and if you're trying to do a de novo rad assembly, I'll just say quickly, we do a reasonable amount of de novo rad assemblies, and I want to make a plug for an assembler called Price. We use an assembler called Price to extend rad contigs, and we're very comfortable with it, and we think it works great for extending rad contigs. So we basically identify single-end rad loci, and use Price to extend them with paired end data. I'll also say this, there's so many things to possibly talk about that, it's hard to know what to mention, but one of the things in general I've noticed is our results are a lot better when we align to a reference genome and then when we do a rad de novo assembly and align to a rad de novo assembly. So even if we have a relatively diverged reference genome, for example, if we're analyzing Chinook salmon, rainbow trout, there's a reference genome for rainbow trout, they're 15 million years diverged, 10 to 15 million years diverged. We have better results by aligning Chinook salmon rad data to the rainbow trout genome than we do by doing a de novo assembly of Chinook salmon and doing that analysis. So as long as your divergence is not too great such that you get high alignment rates, in our experience, the results are better in a number of ways by aligning to a reference genome as opposed to a de novo assembly. So I would highly recommend trying, if you're working on a species and there's a genome that's somewhat closely related, maybe a species in the same genus or something, try that. You can look at the rates of alignment of your reads, and if they're relatively high, for example, if we align Chinook salmon reads to a rainbow trout genome, the alignment rates aren't much lower than if we align rainbow trout reads to the rainbow trout genome, right? So we can, that's a really easy way to get a feel for how well it would work to do a cross-species alignment. And so I think that's, I'm just going to plug in here. I would highly recommend that okay, let me just, a couple, so this is a schematic of this, why did that not get fixed? Okay. I don't want to go over this. Let's look at these data. First of all, does anybody, Gordon or Tiago, do you guys know why this is yellow? And can you fix it?
UC Davis or something?
What?
UC Davis computers?
I'm thinking that that's not the situation. But this is just an example of one of our main justification for developing this new RAD protocol. Basically, we can quantify the number of mapped fragments, or here's a better way to think about it. For every individual, you're going to try this? Okay. For every individual, we have some particular number of sequence reads. Right? So one individual might have 2 million fragments sequenced, another individual might have 1 million fragments sequenced. And we can quantify the number of loci that are covered, in this case, nice.
So that also should say for a bit.
We can quantify the number of loci that are covered greater than or equal to 4x, okay? And so in this case, we were comparing two protocols, this new RAD protocol we presented in this paper and this traditional RAD protocol. And what you can see is that for a particular number of reads, the new RAD protocol actually gives us more loci covered at greater than 4x, but it's not a huge difference, right? But when we remove clones, so we think we have all this we think we have all these loci covered at greater than 4x, which is a reasonable amount of coverage for a lot of things that we do, a lot of the downstream analyses that we use. But it turns out when we actually take these data and then remove clones, in this case, so we thought we had high coverage, but many of those reads were just clonal, and after we removed the clones, now it took, 2.5 million reads to get relatively most of the loci covered, as opposed to 1 million reads, right? So there was a huge shift. So this sort of shows that, and this experiment was actually the exact same DNA, exact same amount of DNA. It's like a plate of DNA just duplicated and then processed with both protocols. So, but there are still some clones. So there's still clones in the new protocol. Does that mean the new protocol always have clones? No, that means in this situation, with this quality and quantity of DNA, and these volumes of the reactions that we did and all this stuff, there's still some clones using the new protocol, okay? Questions about that? This is a comparison of RAD data and Rapture data. So again, when we have RAD data, we isolate all of these tags. In rainbow trout, this is done in rainbow trout, there's about 100,000 RAD tags if we're using SPF1 restriction site. Okay? So if we make a RAD library, we take the whole genome and reduce it to about 100,000 RAD tags. In this paper, what we did is we subsequently took those 100,000 RAD tags and we used Capture to try to reduce them to just 500 RAD tags. And we can evaluate here the number of Rapture loci, so they're defined as Rapture loci if we have a bait to isolate those loci. So the number of Rapture loci covered at greater than 4x given a particular number of fragments from an individual. So if we take RAD data, if we take just straight RAD data, where we don't do the capture step to enrich, if we have 50,000 sequences, 50,000 fragments sequenced in an individual, none of the loci are covered at greater than 4x, like literally none, maybe one or something, just randomly. But if we take that exact same amount of data and we've gone through this subsequent step of further, reducing the complexity and capturing out those specific RAD tags, and then we sequence those, now almost all the loci, these 500 loci, are covered with 50,000 reads, right? Does that make sense? So that's an advantage of you could have done the same experiment where you could have, for example, generated the same amount of data with 2 different enzymes. So the 2 enzymes we use most often are an 8 cutter called SBF1 and a 6 cutter called PST1. Well, it turns out PST1, SBF1 has a site like this, CCTGCAGG, and PST1 is a subset of that. So it's this part. So every PST1 site every SPF1 site is also a PST1 site, but there are more PST1 sites. So you could have done the same experiment and looked at sites that were shared between the 2 enzymes, and you would have saw that you needed 20 times more reads to get the same coverage of those shared sites when you used PST1, something like that. So just in general, you need particular amounts of reads to get particular coverage of different loci, depending on the number of loci that are represented in the sample that you're sequencing, right? Okay, this is the last thing I want to talk about. Then we're going to actually do some computing. So here we're looking at number of SNPs versus position in the Rapture locus. And what we mean by position in the Rapture locus is position away from the cut site. So the distance from the cut site. So the cut site, the first base after the cut site would be position 1. 500 would be 500 bases away from the cut site. So in this situation, in this analysis here, we discovered a lot more SNPs closer to the cut site. So why do we discover more SNPs closer to the cut site?
Coverage there.
Yeah, exactly. It's not that there's the cut site polymorphisms are near SBF1 sites or something like that. It's that because the nature of the shearing step, these second end reads are more dispersed, and the size of the fragments that we shear, the biggest fragments are about 500, and they get smaller. But so we just have less sequence reads out here. And so, for example, less individuals have sequences out there, and we overall detect less SNPs further away from the cut site. So that's sort of the nature of this decline. And so in this case, you might want to know, well, what kind of coverage do I need to get to call genotypes? Well, that depends on how stringent you are in calling genotypes. So what we talked about what we talked about earlier is we calculate posterior probabilities. So if we say there's a polymorphic site, and we're working under the assumption that there's only 2 alleles, so there's 3 possible genotypes. Well, for every individual, we have 3 posterior probabilities. We have the probability that it's one homozygote, given our data, the probability that the individual is heterozygote, given our data, and the probability that the individual is the other homozygote, given the data. Now those 3 posterior probabilities sum to 1, but the question is, well, what confidence do we need to have in a particular genotype before we call it and say this individual has that genotype? So if we decide we're going to make an official call, right? So in this case, we just did a comparison where we said, well, how many individuals were genotyped at each of these SNPs given this cutoff? 80% posterior, 95% posterior, 99% posterior. So which of these is more stringent? What's the appropriate posterior to use when you're calling genotypes? Yeah, it depends, right? It depends how confident you need to be in your genotypes, right? And so to say, like, well, how much coverage do I need? That in and of itself is an insufficient question, because it's how much coverage do you need for a particular question or for a particular analysis done in some particular way. So this is the situation where, what I drew first, where your downstream population genetic analysis is going to influence how you extract genotypic information, which is going to influence how much coverage you generate, that kind of thing. So all of these things are connected. So you have to have some idea, or you should have some idea of what's going on across these steps to be able to optimize your approach.
Mike, what's the sharp drop right at about 100?
Yeah, so what's the sharp drop? So actually, first of all, you see a sharp drop here, but you also see this sharp drop here. Okay? So what does this sharp drop here tell us first, this drop?
Paired end.
Yeah, it just tells us that SNPs that are greater than about 200 bases away, or 250 bases away from the cut site, just don't get genotyped to very many individuals. Okay, so 100% genotyping would have been like 288 individuals. There were 288 individuals in this experiment. However, we weren't very careful in normalizing. Some of those individuals were low, so some of them just dropped out because they didn't have very much DNA to start with, right? So good genotyping is, say, 250 or something, given the care we put into this experiment. So is there some way we could have, say we wanted to cover more, say we wanted to have more SNPs. How could we have manipulated the molecular biology to generate more SNPs? What could we have done to get genotyping out here? What's that?
You don't do a lot of sonications to get longer.
Yeah, so we can manipulate the fragment size. We can also manipulate the coverage. So if we would have got more coverage, we would have had higher coverage out here and more genotypes out here. But then we would have had more coverage unnecessarily in here probably. But what's another way? There's one more way. What?
Reduce the number of individuals.
Well, that gives us more coverage, so more sequences per individual. So we can increase the number of sequences per individual. We can modify our fragment size, right? Our sonication parameter. What?
You increase the cycles.
You can, this is what I'm looking for, you can use longer reads. So now if we would have used, for example, I think the new kits, there's like paired N 250 reads even on HiSeqs. If we would have used paired N 250 reads and we would have had our average fragment size of 500, we would have got a lot more SNPs given the same enzyme. So you can manipulate, you can change many, many things in here to get different, to interrogate different numbers of SNPs confidently and at different levels and all these things. So, you know, fragment size, sequences per individual, sequence length, all of these things. And those interact with confidence, because you're going to need higher coverage if you're planning on, if you need more confident genotypes and these kinds of things. So all these things interact together. Does anybody have any idea what this line is here? It didn't show up here, but it showed up when we had a 99% posterior probability cutoff. A 99% posterior probability cutoff means we're not going to call a genotype unless at least one of the three posteriors is greater than 0.99, greater than or equal to 0.99. Okay, the posteriors are always going to sum to 1. So if one of them is greater than 0.99, the other ones are definitely less than that. Okay? So what is this line right here?
Is it the forward versus?
Yeah, that's the end. This line is actually at 84 bases. Right here, it drops off at 85. Does anybody have any idea why it drops off at 85? Yeah, it's 100 base pair reads, but there's 16 bases dedicated to cut site and barcode. So those are lopped off. You're not discovering SNPs in the cut site or the barcode, because the barcode is artificial. The cut site, you wouldn't pull down the fragment if there was a SNP in the cut site. Okay? So all these types of things, you see them in the data. They make perfect sense when you understand the molecular biology. So these bases are always read by read 1. These bases are only read by read 2. And so read 2 is usually up here, but sometimes it's up here, and these end up being covered less because sometimes the read 2s are out there. So you can also not only control your average fragment size, you can control the tightness of your fragment size too, using gel extractions or you know, these fancy automatic gel extraction machines that they have now and stuff like that.
Has anyone tried 250s to see how well it does with Brad?
Yeah, we've done 250s.
And do you get even all the way?
We've done 250s on MySeqs, we've done 300s on MySeqs. It depends on the quality. Yeah, it would be better than this, but when we did 250s, the quality kind of dropped off. We haven't done it for probably a year, something like that. But it all, you know, for me, we usually would be moving, if we wanted a lot more, you know, this is like a 20-fold step in the number of polymorphisms that we interrogate. So, you know, it kind of depends how many, we don't care about 2-fold changes very often. So we're like here, 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 depending on what we want. We don't manipulate too many of these little things that would maybe produce, you know, 2-fold changes in the number of sites that we interrogate.
Yeah, so this is probably going to be a very naive question, but if you're looking at a large number of SNPs that are very close to your cut site, that's where most of your, looks like return, if you will, in data.
Yep.
But the rate of recombination is going to be really low between them.
Yep.
Are you not violating sort of some
Yeah, so
independence?
let me,